Hello everyone, we are going to cover DNA, RNA, and protein synthesis notes, so jump on in, it's quite a bit. All right, so nucleic acids, these are the macromolecule that holds our genetic material. We call that material DNA. These contain the genes, which are sections of DNA, as it shows right here. It's pointing to a section, and those genes are blueprints for making proteins. They're also located at certain points on the chromosome, and these proteins carry out all cellular activity. So there are two types of nucleic acids. We have DNA, which is deoxyribonucleic acid, and we have RNA, which is ribonucleic acid. Notice the structure. One's a double helix, one's a single strand. Nucleotides are the monomer, if you remember, that make up nucleic acids. They have three parts. They have the sugar, which is the deoxyribose in DNA and ribose in RNA, and this is represented by this little house here. That's the sugar. We have the phosphate, which is represented by this circle, and we have the nitrogen base, which is represented by this green rectangle, and that base is either going to be adenine, guanine, cytosine, thymine in DNA, and uracil in RNA. DNA structure is a double helix. We call it a twisted ladder. The sugar and phosphate form the sugar-phosphate backbone, which is right here. Here's our backbone. Nitrogen bases bond in the middle with a weak hydrogen bond, so right here in the middle, and then all other bonds are strong covalent bonds. RNA structure then is a single strand. It's made up of a ribose and a phosphate backbone. The nitrogenous bases in this one, though, are adenine, cytosine, guanine, and instead of thymine, it's uracil, as you can see from the visual over here. So nitrogen bases bond only with their complementary base pair with hydrogen bonds. So A will always bond with T, apples in the tree. G will always bond with C, so cars in the garage, and that is how it works. So DNA versus RNA, let's do a little recap. The types of nitrogen bases in DNA is ATCG. In RNA, it's AUCG. The type of sugar used is deoxyribose, and in RNA, it's ribose. The shape of the DNA is double helix, while the shape of the RNA is a single strand. So this is a great way to remember. Chromosomes are the suitcases. In these suitcases, we have tightly coiled strands of DNA. One can contain thousands of genes. So think about this suitcase containing thousands of genes. And this long strand of genes are DNA strands. The genes are just one section or one pair of the genes that actually code for a protein. And this is happening in the cell or in the nucleus of the cell. So here's another visual you can look at. We have the chromosomes in here. When they unravel outside of the nucleus, you can see that they are wrapped around little histones, which are proteins. And as they unravel even more, you can see that the double helix shape, and in between here, you can see the nitrogen bases A, T, and C, and G. So DNA replication, it happens during S phase of interphase, when the cell is ready to divide. First, we unzip the DNA. Second, the enzymes help find complementary base pairs to bond with. And third, two identical molecules are formed with an old and new strand, just like it shows in the ladders over here. This ensures that each copy has exact DNA. It's semi-conservative replication because part of the molecule is actually conserved or saved. As you can see over here, half of it's saved and then a new part is coming in. So some questions to ponder really quickly. What kinds of mistakes could occur during this process? And what would be the implications of these kinds of mistakes happening? For example, what would be the difference in a mistake in the DNA of a body cell, such as a brain cell, versus a mistake in the DNA of a germ cell that would later eventually make a sperm cell? Or B, what would be the difference in a mistake if it happened in the gene versus in the chromosome? Think about that. So protein synthesis, this is the process of reading the instructions in DNA to make a polypeptide. We want to make a polypeptide, which is a chain of amino acids. These can bind to others and fold into proteins. As you can see over here, we have primary structure, secondary, tertiary, and quaternary structure. And remember, proteins run our bodies. So from DNA to RNA, it's called transcription, and from RNA to the protein, it's called translation. DNA are the instructions located in the nucleus, which it can't leave, but proteins are made in the ribosomes. So protein synthesis takes 2 steps. First, transcription inside the DNA in the nucleus, and then second, translation with the ribosome in the cytoplasm. So we're going to go over the types of RNA really quickly to remind you what they do. The mRNA is the messenger RNA. These carry copies of instructions. These are carried to the ribosomes in the cytoplasm. It is this complementary single strand. The tRNA is the transfer RNA, which binds and carries specific amino acids to the ribosome. Think of these as little taxis. So tRNA can be taxi RNA, which carries the anticodon to the complementary codons. The rRNA stands for ribosomal RNA, and along with proteins, these make up the ribosome. They also help catalyze the formation of the polypeptide bonds, or the peptide bonds in the polypeptide. So let's go over this quickly. Transcription from DNA to mRNA. The purpose is to carry the code or instructions out of the nucleus. Remember, DNA never leaves the nucleus, and proteins are made in the cytoplasm by the ribosomes. The location of transcription is in the nucleus. It starts with DNA and it ends with mRNA. Transcription, we unzip the gene that needs to be copied. We use complementary base pairing rules to match the RNA nucleotides with the exposed DNA nucleotides. Remember, RNA has uracil, so A will bind with U and C will still bind with G. Then we release the completed mRNA molecule, and DNA zips back up and the mRNA leaves the nucleus and enters the cytoplasm. Here's a visual of this. So we have the DNA that unzips. It uses mRNA to make a complementary base pair, or to make an mRNA, that is. That mRNA is released and the DNA zips back up and the mRNA is allowed to leave the nucleus. So here's an example of a DNA sequence template and the mRNA sequence that goes with it. If you want to pause and check this out, this is a good time to do that. So here's some vocabulary that you need to remember. Genetic code is the code of instructions for how to make proteins. The codon is a set of 3 nucleotides on the mRNA. And then the transfer RNA is the anticodon, which is complementary, 3 nucleotides on the tRNA. And this little guy up here is the amino acid monomer building block for making proteins, which are held together by peptide bonds, which will make a long strand of polypeptides. So going into translation now, this is from the mRNA to the polypeptide, or the protein. So the purpose is to read and follow the instructions carried on the mRNA to make a polypeptide. The location is in the ribosomes. It starts with the mRNA and ends with a polypeptide. So here's the process. Number 1, mRNA attaches to a ribosome. Number 2, the ribosome reads the mRNA codons, starting always with AUG. So 1 codon is equal to 3 RNA nucleotides. 3, the tRNAs act like little taxis to pick up and drop off the amino acids that match with each codon. And then number 4, the tRNAs continue to drop off the amino acids, and the ribosome binds the AA, or the amino acid, together with the peptide bonds. And then finally, when the stop codon is reached, the ribosome releases the completed polypeptide chain. So the translation is the interpreting of the RNA message into a polypeptide to make a protein. That's the whole purpose. So here is a genetic code chart to find the amino acids. I'm just going to help you with the first one because it always starts with AUG. So you start from the center and you have A, then you find the U over here, and then you find the G right here. So AUG will always be methionine. So here are some examples. We have a DNA sequence and then an mRNA sequence that matches up with the base pairing rules. Then we have the codons, which are just taking these apart from the AUG first to the next three, to the next three, and so forth. Jump down to the anticodons. These are going to help you find what the amino acids are. So these are the codons that come in and will create that the amino acid on the polypeptide chain. And then these amino acids are going to be what those are made of. So that we always write the first three letters of the amino acids, MET, ARG, and so forth. Okay, so our summary really quickly. The purpose of DNA replication is to make identical DNA strands during S phase and interphase. The building blocks are A, T, C, and G, the nucleotides, and it happens in the nucleus. Transcription is to produce a strand of mRNA from DNA, and this happens in with the nucleotides A, U, C, and G in the nucleus. And then finally, translation is to produce a protein from mRNA. The building blocks are tRNAs and amino acids, and the location is in the ribosomes. Great job today, you guys. I hope this made sense and it was helpful.